Hey guys, welcome. This is just a review for the last day of the study guide in case you were unable to attend the class today, or we only had one odd day this week. So in case you're in the odd day class, sorry for that, but we're going to have to go ahead and do this way rather than meeting in class because we just literally don't have time. So the first thing you'll need to do, of course, is to go to the 4.7.4 midterm study guide. That is where the study guide lives. No matter what, we're going to always come back to this. So you're going to scroll down and click on the study guide to pull it up in another tab. All right, and then once you got that up in another tab, we're going to go back to modules, and we're going to go all the way down to today's review. Now, when we do this, it's going to go back to ecology and everything else like that, which is cool, right? So lesson 9, midterm review, part 3 is what we're looking for. We click on the first one, back to the beginning. It just tells us that we need to make sure that we pull this up and reminds us that this midterm study guide is a test grade, okay? So make sure that you turn this in. So once we're done today, I will go ahead and show you, but once we're done today, you are going to make sure that you turn this in. Go back to 4.7.4 and press the submit button once you are done filling out this student copy. Okay, so we did a quick practice problem before we begin. What type of macromolecule would you want to consume the most before heading out into the cold wilderness to survive for a long period of time? I'll give you a second to try to answer it. Okay, the answer was lipid, because lipids perform long-term energy. They provide for long-term energy. So lipid provides energy and insulation. We're going to learn a little bit more about that today. So the subjects, the topics we're going to cover are all of these, from the scientific method through some general ecology and through some macromolecular function, and ending with enzymes. Okay, so we start with the scientific method review, and basically the scientific method review goes through all the different pieces of scientific method. Experimentation is how we basically go about testing the hypotheses we come out with, and our hypotheses are essentially our explanations of what we think reality is. And of course, we go off and test this notion of reality to see whether or not reality actually behaves the way we would expect it to do so. And that's what we call experimentation, is when we go out and test that. Now, your experimentation can say, yes, either your version of reality is pretty close, or it could be that it's not accurate at all, and that's okay. It's fine when you have a hypothesis that doesn't fit what you think should happen. A theory is a little bit different. A theory is a hypothesis that's supported by data, so it's basically fact. So when we talk about things like, the theory of evolution, it's not just a theory when people say that. It's essentially fact. It's been proven by many, many experiments. Evolution is a thing, and when people say, well, why don't you just make it a law? Well, the reason we don't make it a law is because scientists have been burned in the past about saying that they understood everything about something, and so we typically tend to be very careful. And we say it's a theory because it's fact, but there's still some things that we might be able to learn about it that would change our perspective on it a bit, okay? So basically we could have a more nuanced view with more data. So we're always allowing for our view of the world to change, and that is an important part of science and life. Okay, so in experiment, we start with a hypothesis, which is an argument about the world that predicts what will happen when we change it in a certain way. The independent variable is the thing that we specifically change. The dependent variable is what we're looking for to change. The control is going to be what would have happened if we hadn't changed anything. So basically, we want to see whether or not the change we made actually did something. Our constants are going to be the things that we keep the same. So you want to make sure that you're only changing one thing at a time to make sure that other things aren't also affecting it. So everything else is a constant. We collect data based off the experiment, so you look at the way the world reacted, you make a graph or some other statistical analysis or something to analyze the data that you got, and you come to a conclusion about how accurate of your hypothesis was. So whatever you do, only test one independent variable at a time, no matter what, and then also make sure that you have a pretty good control, because otherwise your independent variable might not actually have changed much at all. So now that we have gone through this, you basically, down at the bottom it says, now that you reviewed the scientific method, please complete question 1 in your study guide. So we're going to go to question 1. Okay, and the data set below was taken by a scientist trying to see how fast people's reaction times were when somebody jumped out to scare them. So we have their trials and the time to reaction. All right, then we're going to ask you a couple of questions. Do any of these data points stand out as being different from the rest? Okay, so for this one, what I would suggest doing, I'm actually going to give you a second to pause the video and try to do number 1, A through C, and then after a second or two, once you're done, come back over here and check it. Okay, hopefully at this point you pause the video and you've already done these, so let's go over them. Do any of these data points stand out to you as being different from the rest? Yeah, absolutely. If we take a look at this right here, 1 second, 1.5 seconds are pretty close to each other, but 10 seconds is pretty long, so that's weird. Originally, the scientists just wanted to do one trial of the experiment, but his friends convinced him to do more trials. Why was it important that he do that? Well, something funky happened here with the first trial. If he had only done the first trial, he might think it takes 10 seconds to react to something scary, which, think about how long 10 seconds is when you're really thinking about it. I mean, if it was something dangerous, you'd be dead in 10 seconds, right? So that wouldn't be great. So if he'd just gone off the first trial, he would have bad information because he would think that this was how long it took. So it's really good to do more trials to make sure you have an accurate idea what's happening. To make the scientists' experiment even more accurate, what might you recommend them do? Well, definitely more trials. Definitely more trials to make sure that they're more accurate, because who knows, this might go back up again in time. The more trials you have, the more accurate a view that you have. Okay, so that's how you do that, and then you go on to the next section, and the next section is about trophic levels. So I'm not going to go through the rest of this. I'm sure you guys can read, and you can go through and pick out all the stuff. I bolded the really important information and highlighted the super important information and all that stuff. So you should be good to go. But I just want to point out to you, so you're going to read through the overview and the trophic levels. You're going to come down here to the bottom, and you're going to notice it says, now please complete questions 3 through 6 and 8 through 10 in your midterm study guides. Basically, you're going to come down to these sections and do that. Go through symbiosis, now complete problem 2, go through succession, now complete problem 7. You get the idea. Okay, so you're going to go through and complete different things in your study guide. I'm going to go ahead and go over those, each one, and please pause the video before I do and try it yourself first, because that is the best way to learn. If you try and then you still fail, and you see that, hey, my version of going over is very different than yours, at least you have an idea of what you need to work on. You can go, that's what I got wrong. I'll remember that for the test. That's really, really helpful. All right, so let's start here with 3 through 6 and 8 through 10. Okay, so let's take a look at 3 through 6. All right, please pause the video at this time and try these after you've gone over the section, and then we'll go over it together. Okay, 3. List the decomposers shown in the picture above. Now, decomposers are going to be things that are going to be receiving the basically dead or decomposing matter from organisms, after they die. So, right, we're going to see the mushroom is going to be an important decomposer. The earthworm is going to be an important decomposer, and bacteria is going to be an important decomposer. So anything basically that's in the soil or down here, it's going to be breaking down dead organisms are decomposers. If the fox population increases, what will happen to the rabbit population? Well, the foxes eat the rabbits. So if more foxes are around, the rabbit population will go down. If the fox population decreases, what will happen to rabbits? Well, since the foxes eat the rabbits, if the foxes go down, the rabbits will go up. If the rabbit population is completely removed from the ecosystem, will it affect other species? Absolutely. So the rabbits eat the grass. If the rabbits are all gone, the grass could grow up to huge proportions, right? If the grass grew up to huge proportions, it might kill the mushroom or make it so that there wasn't, I don't know, maybe more shelter or something for something else to move in. It could change the bank and the way that the bank is, and how much water was able to get in. The other thing, though, is without the rabbits, the foxes wouldn't be able to support themselves, right? And since these birds feed off the foxes, then they wouldn't be able to support themselves. And then since this eagle also feeds off the fox, excuse me, this fox eats the bird, so that these birds would actually grow larger, and this bird would die off because it didn't have foxes. So basically, the whole point is it's all interconnected no matter what, and you have to make sure you look at the arrows to see who eats who. The arrows point towards the thing that's doing the eating, right? The higher level predator. So this fox gets eaten by this bird, these birds get eaten by this fox, so do the rabbits, etc. Okay. All right, cool. So that's 3 through 6, and then 8 through 10. Oysters are keystone species in the Chesapeake Bay. If other oyster populations decline, the water quality will decline. This will be detrimental to many aquatic species, causing a lack of essential resources. So what do oysters do? They're filter feeders. So oysters are going to remove toxins from the water. They're also going to help to increase the clarity of the water and make it easier for plants to do photosynthesis, so there's more oxygen in the water. So basically, once they're removed, there'll be less oxygen in the water, there'll be more toxins, the water will be dirtier, and it won't be nearly as healthy. So those are some things. And then, of course, they're going to affect other species as well because they'll be affected by the poison, etc. Scientists have been studying a forest for the past 20 years and determined the ecosystem to be stable. Recently, there's been an increase in the wolf population, and now it exceeds carrying capacity. How does the increase in the wolf population affect the stability of the forest ecosystem? Okay, so the wolf population is exceeding carrying capacity. So essentially, at this point, if you remember, the carrying capacity is the total amount of basically space and resources that can support an organism. If it's gone over that, then the wolf population is going to become unstable. So the ecosystem becomes unstable because it's exceeded its capacity. Once it goes back down underneath it, it'll probably stabilize. Label each organism's role in the food chain below using the following terms: primary consumer, secondary consumer, producer, tertiary consumer. Okay, well, the grass is a producer because it makes its own food. The grasshopper, because the grass is the primary or first consumer that feeds on the grass. The frog is a secondary consumer because it feeds on the primary consumer, and the python is a tertiary consumer because it feeds on the frog. So tertiary to frog, you get the idea. Okay, so hopefully you paused the video before you went through all those, and now let's go ahead and go to the next one, which is after symbiosis, problem number 2. Okay, go ahead and take a second to do that. Pause the video. Okay, for 2, only use the words listed below to complete the following paragraphs. So symbiosis, mutualism, parasitic, and commensal. Okay, blank is a close relationship between species involving survival, which results in either of these 3. That's symbiosis. Okay, crustose lichens are made up of fungi and algae. They work together as partners while breaking down rock. Fungi help protect the algae provide nutrients to the fungi, so they're both being helped. So this one is mutualism. In some relationships, an organism can harm another organism while reaping the benefits. And these, in humans, these blank relationships lead to malnutrition, jaundice, diarrhea. So this is parasitism. Whenever one organism is being hurt, the other is being helped. It's parasitism. And then finally, spiders are known for the elaborate spider webs. When a spider creates a web between the antlers of a deer, is able to catch food as the deer travels. Their relationship is a commensal relationship because the spider benefits if the deer is neither hurt nor harmed. Okay, so basically in commensal relationships, one organism is being helped and the other one really doesn't care. Okay, and now we're going to do problem 7. So go ahead and pause the video. Okay, Hawaii's most active volcano, Kilauea, that's cool, had its last major volcanic eruption in 2014 and lasted for several months. So the volcanic eruption covered the entire island, leaving only volcanic rock on the island. Which type of succession will occur? What pioneer species are needed? Okay, so volcanic rock, if it's starting from rock, no matter what happens, if it's starting from rock, it's primary. Okay, so I know you might be confused because you're like, well, the volcano is a natural disaster and it killed everything, it should be secondary. Well, the rock is a big thing. If you have no soil, it can't be secondary, even if there was some natural disaster that caused that. So you have to have soil to be secondary. Okay, primary is when you start with rock. So primary is the type of succession. What pioneer species are needed? Well, remember, pioneer species are needed to break down rock in most cases. So lichens, mosses, and grass break down rock. Those would all be good for that. Okay, so now let's go ahead and move on to the next section, and just like before, you're going to be reading through water properties and all that stuff, and at the very bottom of this, after you read through the different water properties, 11 through 16. So let's go to 11 through 16, go ahead and pause the video. Okay, now we're going to go through 11 through 16. So, either cohesion or adhesion refers to water molecules sticking to water molecules. If they're sticking to each other, then it's cohesion. It's adhesion when they're sticking to other molecules. So when you're adding an additional thing, that is adhesion. Okay, co means together, so when water molecules are together, sticking together is cohesion. Give 3 examples of adhesion. Anytime water has stuck to something is adhesion. Okay, so water on the windows in the morning when you get up. Water on the grass, right? The dew on the grass. I'm trying to think of others, and somehow my brain is completely blanking. Water on the windshield, water on the sides of your shower. Anytime that you see water sticking to something, that's adhesion. Give an example of water helping living organisms because of its high heat of vaporization. Okay, this basically means that it takes a lot of energy to heat up water enough to be able to evaporate. So a great way that water helps organisms through this is through sweat. So when an organism sweats, because the water on the surface of their skin can absorb so much heat before it evaporates, it can actually suck the heat out of their body in order to cool them down. So that's how sweat helps them cool down. We call that evaporative cooling, and it's a great thing. Give an example of water helping living organisms because of its high specific heat. Well, this is another way in which that's important. But also, because water needs to reach like a pretty high amount of energy in order to actually start boiling or even heating up at all, it's really, really good to use as a substance for living things. So basically to keep your body temperature cool, even with all your enzymatic reactions and stuff. But another way that it can be really helpful is to regulate the climate of the Earth. So it takes a lot of heat to heat water up. So basically, water can absorb a lot of the heat from the atmosphere and make the Earth a lot cooler. How about its ability to be universal solvent? Well, that's really useful for living organisms because guess what? Your blood is like a high water-based solution, and you need to carry a lot of stuff through your blood. And so, because water is a great universal solvent, you can dissolve a lot of really important things like sugars and certain proteins and stuff in your blood, and it can help to transport them to different parts of your body. Sorry, my brain is just totally fried, so I really apologize for this, guys. And then give an example of water helping living organisms because of its low density as a solid. So because it's less dense when it freezes, water becomes ice that floats on top of other water. And that's really important because it will actually insulate the water beneath it. So it makes sure that like when a pond freezes, for example, and the pond is full of fish, the whole water doesn't freeze like under it because it's insulated by the sheet of ice on the top. So the fish all live, basically. So, okay, all right, so that's the water properties. Whew, my goodness, rolling right along. Now we get to macromolecules. So please review this for the macromolecules, and then you will see at the bottom of all this, please complete 17 through 23 in the study guide. Whew. All right, 17 through 23 in the study guide. So go ahead and take a moment to pause the video. And once you have paused the video, let's go ahead and begin. So DNA contains instructions to produce blank, the building blocks of proteins. The building blocks of proteins are amino acids. Fatty acids and glycerol heads can be combined to produce essential for making cell membranes. Those are lipids. The monomer of nucleic acids is the nucleotide, composed of phosphate, sugar, and nitrogenous base, so nucleotide. A single sugar monomer is called a monosaccharide. Mono means one, saccharide is sugar, saccharine, sugar, monosaccharide. Let's see. Responsible for catalyzing almost all chemical reactions in the body. Enzymes are a special form of protein that are required for our survival, and then essential for rapid energy and also for structural components like cellulose in the cell walls of plants. Carbohydrates are commonly found in sugary candies and starchy food like spaghetti. Then 23, when animals such as bears hibernate, they're not getting nutrients from food for long periods of time, and they rely on stores of molecules in their tissues. Which molecule provides energy for the body? Which molecule would a bear use up quickly? Which molecule would a bear's body rely on to provide energy long term? Good God. Okay, well, so let's break this down. Which molecules provide energy for the body? Well, carbs and lipids both provide energy for the body. Carbohydrates are like fast-term, short-term energy, and lipids are long-term. So which molecule would a bear use up quickly? It would use up its carbohydrates very quickly and burn those very quickly to get energy. And then which molecule would the bear's body rely on to provide long-term energy? That would be fats, lipids, because they are very slow burn. Okay, guys, and then, oh my goodness gracious. Okay, so there's one more page. Actually, no, wait, sorry, enzymes. I skipped enzymes. Sorry, so enzymes, and, oh, that's why. I'm so sorry, guys. I forgot to put a little thing. I'll fix this before you actually do it. But there should be another little thing here, right here, that tells you to finish problems 24 through 28, excuse me. So, I'll put that in before you guys see it. God, this is terrible. Anyway, so based on the graph below, what is the ideal pH range for the enzyme pepsin, and which is ideal for trypsin? Okay, well, if we look at this, the rate of the enzyme is highest for pepsin at about 3, and for trypsin at about 8. So that would be my answers for those. Then, oops, sorry, pause the video. Oh God, pause the video if you haven't already, for 25 through 28. And let's continue. What happens to an enzyme's active site if it's placed in an environment that pushes it too far out of its tolerable range of pH or temperature? What is it we call this process? Well, we call it denaturing. Haha, denaturing. And it unfolds the protein and makes it so it can't work anymore. Okay, 26. Please indicate which of the following ways that enzyme activity can be decreased. Okay, if we have more substrate concentration, that's going to actually increase our reaction rather than decrease it, so no. Denatured active site is going to decrease it. Okay, so that's the answer. Higher enzyme concentration is not going to decrease it, and extreme pH or temperature is not going to decrease it, so denatured is the right answer. 27, please indicate which of the following ways that enzyme activity can be increased. Okay, well, this one, yes, for sure. Yes, and then not this one down here. So this one, so sorry. Oh, I see. There's more than one answer. I apologize, guys. I'm so sorry. So these two up here would decrease it, and then these two would increase it. All right. So substrate concentration is high, enzyme concentration is high. That would increase it. Denatured active site, extreme pH or temperature, that would decrease it. Okay. The graph below shows the activity level of the enzyme cholinesterase, an enzyme that helps with neural cells in the brain. Relative activity, here it is. Okay, and then what is the best reason to explain why the graph levels out at 6 minutes? Okay, at this point, notice that the reaction is not, it's not shutting down, but it hasn't, it's not happening any faster or slower. So it's basically reached like a maximum amount. So it's reached the maximum capacity. Okay, if you increase the substrate concentration, it will continue to rise. If the temperature of the solution had been lowered, it would literally go down. This line would go down because it would stop actually doing anything, and then the reactants have been removed would also cause the line to go down to 0. But because the line is remaining steady, there is still some reaction happening. It's just not be able to happen any faster, so it's reached the maximum capacity. Woo, okay guys, that was the last section. I'm sorry this is so long, but hopefully that was helpful for you guys. So thank you so much for putting up with my craziness and being patient. My brain is pretty fried, but I hope this is helpful. Let me know if you have any questions. Just email me, and I'll get back to you soon. All right, take care, guys.